Good afternoon. I'm one of the biology tutors over at the STEM Center, and I'm here to talk about Punnett Square. Now, the reason why we like to use punnets in biology is to look at genetics. In genetics, we like to look at things phenotypes and genotypes. Now, the word genotype you might relate to genes, and what our genes are traits that kind of say what we are even. So genotypes relates to our genes, the genetic material that we get from our parents. One chromosome from our mom, one chromosome from our dad. Now, our phenotypes, think face. So what do we show? What it's the expression of the genes. So, well, my phenotype might be brown hair. My genotype could be brown dominant, red recessive. And so I want to show you the difference. Go ahead and share my screen. Okay. And so when we talk about it, let's go to eye color. So we have our parents' eyes. Let's say one was brown dominant, and we had our mother's eye. So let's say parent one. This is for eye color. Parent two was recessive. Okay. Now going back. So let's say parent two. Now we want to know what kind of child they would produce. Well, what we do is we like to utilize our Punnett squares. This would be our genotype. And we would say that our genotype is homo, meaning one. So zygous. And dominant. I'm not an English major. I'm a biologist. That's wrong spelling, then you can fix it. Well, this genotype for parent 2 is also one type. So homozygous. And because it's our lowercase, let's say recessive. So homozygous recessive. Alrighty. So we want to know what kind of child they would produce. If we have a homozygous dominant and a homozygous recessive. So what we do is make a Punnett square. And to do that, you build a square. put little crosses through. It's a window. We're looking into how or what these parents produce. So I like to put all of my capitals on top. You may have seen something like this in your math class. Okay. So I put parent 1 on the top and parent 2 on the side. Now we want to know what their offspring is. So we bring down, it's always going to be our dominant case first. And so our dominant goes down and we put them in 1 on each. So B, we put a, goes all the way down, capital B, capital B, capital B. And then you follow with your lowercase. And to see how many, what type of child they have, well, then we count up the different varieties. Here we only have 1 variety, right? All of these are the same. So we would say 4 of them are, and we'd say, well, out of 4 times 100% would give us 100%. Well, notice how these two are different. When we say different, we say hetero. We would say this is heterozygous. And because of this capital, this dominant gene, we would say it's heterozygous dominant. So recap. We wanted to know what are the child of 2 different parents. And we utilize our Punnett Square to figure this out. If we know the genotypes of each parent, then we can figure out the probability of the genotype of their offspring. Knowing the genotypes can also tell us the phenotypes. Now remember, genotype is the genes that we carry, the genetic code. Well, phenotype is the face we wear. So pheno, face, it's not spelled it, so I should probably spell pheno. So phenotype, is the face we wear. So phenotype, I like to say the face we wear. And so the phenotype here would be brown dominant. And the phenotype here would be blue recessive. because we're just using eye color. Okay, so brown dominant, blue recessive. Now, what happens when we have a heterozygous dominant offspring or parent? Well, we say that the dominant, that capital, overpowers the recessive. And so we would say that this phenotype is brown-eyed dominant. Now, there are exceptions to the rule. Science has taught us that there's almost always exceptions or differences in the rules, but this is your basis. This is going to be your base for utilizing Punnett squares in figuring out phenotypes and genotypes if you're given two parents with different genotypes. Now, you can also utilize this method to find out the genotype and phenotype of offspring if your parents had the same or multiple generations of genotypes. So as long as you have some information, you can solve for this. So let's say we worked with a blood type. And this one is very common. Let's say that offspring 1 so we're going to say that some famous dude or lady had an offspring. And offspring, just say it offspring number 1 and offspring number 2. And these two children had the blood type. They were both O blood type. O blood type. And the mother was doing a paternity test to figure out if the father was the father of the offspring. And so we know the mother's blood type to be type A. And we know that the father's blood type is type AB. So the question is, are these children these children the father's children? Well, to figure this out, we have to first figure out, well, what kind of children can the father and mother produce? We know that of the 3 blood types, there are 4 different types of blood types. Excuse me, not 3, 4. We have, and we like to say I for blood. So for dominant, we have 2 different types of dominant genes. We have the A and B. And then our O blood type is actually a recessive gene. And we use lowercase because it's recessive. Now these 2 types, these 2 are what we would say codominant. Meaning that both of them will be expressed if present. Okay. This is our recessive. Okay. So if we have two codominant genes and one recessive gene, we know the blood type of the mother and blood type of the father. Can we say that the blood type of the offspring are belong to the father? All right, so we look at the father's blood type. And his possible blood type, well, he has two that are being expressed already, which means that his blood type has to be IA and IB. While the mother's, on the other hand, she could be homozygous dominant, meaning IA, or heterozygous dominant, meaning capital IA and lowercase I. We know that the recessive carriers and recessive genes are going to give us our O-type blood. Okay, which means that the offspring right here both have to have lowercase I, lowercase I. Now, we know that they'll get one from their mother and one from the father. So by process of elimination, we can immediately cancel this out. The reason why is because the mom will have to donate one recessive gene. Okay. So for that to be true, we ask ourselves, are there any recessive genes here? The answer is no. Therefore, these children cannot be the children of this father. And to prove that, we can use our Punnett square. If the two were to copulate, then we would say that the father has AB and the mother, oops, should use the I notation, huh? The mother has to have AI. Okay. Remember, we bring our capitals down. This goes down. Okay, make them crisscross applesauce. This one, A before B, right? And capital before lowercase. And so the possibility of these blood types, if the two were to copulate, would be a variety. 1 out of 4 homozygous dominant. 1 out of 4 heterozygous codominant. 1 out of 4 heterozygous dominant. And another 1 out of 4 heterozygous codominant. And so the phenotypes, the what's expressed would be A, B, A, and B. There are no type O bloods. Therefore, these offspring cannot be the children. And that's our base work for Punnett squares. If you have any more questions, feel free to ask. Or you can always message me directly on Teams. Thank you.